The topic for my talk today is phenotyping microbial cells, how and why. So let's start with the why question. Phenotyping allows one to determine properties of microbes and how those properties change as a consequence of genetic, epigenetic, and environmental alterations. And throughout my talk, I'll be illustrating this with numerous examples, covering characterization and culture of microorganisms, gene function and genomics, environmental effects, and pathogenesis and epidemiology. How to phenotype is a more difficult question, because in the past, microbiologists have tested phenotypes very inefficiently one at a time, usually by streaking or spotting cells on agar plates. So to make phenotyping powerful and practical, what was needed was a high-throughput phenotyping technology, which is called Phenomics. This slide illustrates information flow in cells, starting with DNA and then to RNA and then to protein, and eventually resulting in cellular properties or phenotypes. A lot of cell analysis is done using the molecular analytical tools, molecular omics such as transcriptomics and proteomics. And now we have available another option, Phenomics, which is a live cell analysis. With the molecular analyses, one measures levels of RNAs or proteins and then extrapolates or guesses as to how that will result in biological changes in the cell. One of the big advantages of Phenomics is that it's measuring at a higher level, at the cellular level, where you're actually seeing what important changes are occurring in the living cells. Another major advantage of Phenomics is that because it's a live cell measurement, you can set up one experiment and follow the cells over a period of hours to days. Whereas with the molecular tools, you have to do discrete sampling and do a time course experiment. So this is a typical wall poster or a picture, or what I think is a typical image that we all have in our brain of what's going on in the cell, a cell model, so to speak. And cells have lots of pathways that are interacting, and it's just incredibly complicated. And at first blush, it seems like an impossible task to try to make measurements of the various pathways that result in cell phenotypes. But actually, we've come up with a very simple way of doing this based on a simple assay principle, which is a colorimetric analysis of energy production. And to explain this, I need to have a simpler model of a cell shown here. This shows only three pathways of the cell, but they happen to be three particularly important or central pathways. In the center is the electron transport chain of a cell, and this is flanked by the glycolytic pathway on one side and the TCA cycle on the other side. And so with this simplified view of a cell in mind, we can do start to do very simple phenotyping by just simply feeding the cells one chemical at a time and measuring the energy that the cells are producing. So for example, if we feed the cell the amino acid glutamine, first glutamine has to be transported across the membrane to get into the cell. Then it's converted in a few steps to alpha-ketoglutarate. Alpha-ketoglutarate dehydrogenases, removes an electron, transfers it onto NAD to make NADH. And then that electron can go down the electron transport chain and onto one of our redox dyes, forming a purple color. So we can make an in vivo measurement of the flux through this pathway by simply feeding the cells glutamine and measuring the rate of purple color formation. And then in a second test, we can feed the cell a different chemical, for example, a sugar like fructose. Fructose enters the cell through different transporters, gets converted to glucose 6-phosphate. Again, an electron goes on to NAD to make NADH, down the transport chain, and again onto our redox dye. So these are two examples of nutrients that have pathways that we can track and measure. And you could imagine feeding the cell a whole wide range of hundreds of nutrients. Each one follows a different pathway to energy production. And by doing all of these measurements simply and easily, we're getting a lot of information about the metabolism or metabolic properties of that particular cell. And again, if you think of the master circuitry of the cell, each individual measurement is a subcircuit, And what we're doing then is by testing many subcircuits of the cell, we're eventually getting a more and more complete picture of the cell's master circuitry. And this is very easy technology to do. The physical state of these assay plates are just 96-well microtiter plates with different nutrient chemicals dried in the bottom of the wells. So in this example, I'm showing one well with fructose, one well with glutamine, other wells with other nutrients. To run the assay, you simply pipette in a cell suspension with the redox dye, and over a period of hours to days, the cells will produce energy differentially in these different wells, and you'll get different rates of purple color formation. And if the well contains a chemical that the cell does not have a pathway for metabolizing, then the well will remain colorless. So you can imagine hundreds of measurements of this type, but then you're probably thinking, okay, I can see how this is measuring the catabolic pathways of a microbial cell, but there are a lot of other pathways in the cell, and how are we going to measure these other pathways? And so we can measure hundreds of thousands of other pathways by using inhibitors, pathway inhibitors. And in this case, we suspend the cells in a rich nutrient medium so that they have many pathways for generating energy. And we now include in the wells inhibitors of specific pathways. So in this slide, I'm showing two examples: oligomycin, a mitochondrial inhibitor in one well, rifamycin, a RNA polymerase inhibitor in another well. And then these are probes of mitochondrial and transcriptional activities in the cell. And now we run these assays the same way with the same redox dye. We add the cells and dye again, and now instead of looking for wells where we see stimulation of purple color, we're looking for wells where we see inhibition of purple color. So the basis of these hundreds or thousands of measurements is a simple redox dye chemistry measuring the energy output of cells. These are pre-dispensed and dried into different microtiter plates. So we have plates with chemicals that stimulate energy production by cells and chemicals that inhibit energy production by cells. The other part of the technology is an instrument called the Omnilog, which simultaneously incubates and records the purple color kinetically so that you can measure rates of purple color formation in hundreds or thousands of assays run simultaneously. For over a decade now, we've had a set of almost 2,000 assays, phenotypic assays, that can measure carbon metabolism pathways of cells, nitrogen metabolism pathways, phosphorus and sulfur pathways, biosynthetic pathway activities, effects of osmolarity and specific ions, pH effects, and then also sensitivity of the cells to 240 selected inhibitory chemicals that block different cellular pathways. And again, these assays are easy to run. It simply involves pipetting in a cell suspension, 100 microliters per well, loading these plates into the Omnilog. The Omnilog reads them every 15 minutes, and at the end of the experiment, you get a kinetic result for each plate and for each well, showing the rate of energy production versus time. So I'd like to talk about specific applications to illustrate the technology. Certain questions that we can address include what are the cell's properties, how can I culture the cell, or how can I culture it better, in which environments will it thrive, how can I selectively enrich for it or selectively kill it if you're trying to isolate a particular organism out of a complex environment, how can you improve the microorganism's use in an industrial bioprocess, and how can you use this technology to improve metabolic modeling of cells. So let's start with an example here. This is a phenotype microarray analysis of an important bacteria called Corynebacterium glutamicum. This is used in very large-scale fermentations to produce glutamic acid and other amino acids. So it's a very industrially and economically, nutritionally important microorganism. By doing a phenotype microarray analysis, we can learn a lot about the properties of this microorganism. The first two plates in the upper left corner measuring carbon metabolism, so we can see a variety of different carbon sources that this microorganism can use, which ones it uses strongly, which ones it uses weakly. Then we have 4 plates for nitrogen metabolism. The top 2 rows of plate number 3 have all 20 amino acids, and we can see that this microorganism uses ammonia and urea, and in terms of amino acids, primarily uses the amide amino acids, glutamine and asparagine. However, even though it doesn't use most single amino acids, it will use amino acids when they're provided in peptide form, as shown in plates 6, 7, and 8. The other interesting thing that I want to point out about Corynebacterium glutamicum is its osmotic resistance. It's very osmotolerant. The top row here in plate number 9 has a titration of sodium chloride from like 1% up to 10 or 12%. And you can see that even at the highest concentration, you see as you increase the salinity or the osmolarity, the metabolic signal energy output decreases gradually, but even at the highest salt concentration, these cells are still able to thrive. Now, this is very important because this microorganism is used to produce sodium glutamate, and in a fermenter, the sodium glutamate concentration, the osmolarity keeps getting higher and higher, but this microorganism can continue to produce it because it's very osmotically tolerant. Now let's contrast that with a very different microorganism that's also an important one. This is a Streptomyces coelicolor, a soil Streptomyces. It's an antibiotic-producing bacteria, so this is also grown industrially and commercially in fermenters. We see that it has a very different profile of carbon source utilization. One of the carbon sources it uses very strongly happens to be gelatin, which is a protein, indicating that it has strong protease capability. Now, if we look at plate number 3 for nitrogen metabolism, we see almost every well is positive, so it can use every amino acid as a nitrogen source, as well as nitrite and urea and ammonia. But it does not use methionine. That's the one exception, and we see here also that it doesn't use peptides containing methionine. So that's the one amino acid that it does not remove nitrogen from. Now, if we look at plate number 9 with the osmotic and salts, we see that in drastic comparison to the Corynebacterium, the Streptomyces does not like salt at all. Even at the lowest salt concentrations, it's mostly inhibited. It will tolerate osmolarity, though, as long as it's non-ionic. So urea it tolerates very well, but ionic compounds tend to be inhibitory. So in one experiment, you get a lot of information. The next example I'd like to show is a beautiful paper from NIH in Rocky Mountain, Montana, where they used Phenotype microarray technology to figure out how to culture a microorganism that had been considered an obligate intracellular pathogen. In other words, it can grow inside of animal cells, but if you break the animal cells open and try to grow the bacteria on a typical agar or liquid medium, the cells would die and not grow. The big advantage of the Phenotype microarray technology is that it's not measuring growth, it's measuring energy production. So even in cells that are not growing or cells that are in the process of dying, we can still make some measurements and get some useful information. So what these scientists did was start by growing the cells, growing the Coxiella inside of a host mammalian cell line. They break the cells open. They know that the bacteria are dying, but very quickly they put them in the Biolog plate with different carbon sources, and they get one positive reaction in the well for succinate. This is indicating that they have the capability of using succinate as a sole carbon source. And so with that in mind, they repeated this experiment, but using a hypoxic chamber where they could control the oxygen tension. They suspected the bacteria was oxygen sensitive. And sure enough, as you titrate down the oxygen level from ambient 20% oxygen down to 2.5% oxygen, you see more and more pathways turn on. And in fact, the microorganism could use a large number, I think 17 different carbon sources, but still succinate, even at the 2.5% oxygen concentration, succinate was the favored carbon source. And then in other experiments, they defined the salts that this microorganism needed. They found out that it's more active metabolically at pH 4.5 than at pH 7, and they found out that it needs high level of cysteine. And so by stepping through these different phenotype microarrays, they could figure out what this microorganism needed in order to grow, and they were able to finally come up with a culture media so that they could grow it outside of the animal cell. That's a major breakthrough. One industrial example provided generously by Nigel Mounsey at Dow AgroScience. He was on a project where they were trying to optimize the titer of a natural product that was produced by some microorganism they were working on. And this shows little growth curves superimposed with the 96 carbon sources of the Biolog Phenotype microarray plate number 1. And you can see some of these carbon sources are used for growth and some are not. Now the colors are indicating, however, what's important, which is the titer of the product that they were trying to produce. And they found a very dramatic induction of the product that they were trying to produce in the well with acetic acid, which happened to be a non-growth condition, but acetic acid was a very good trigger to get the cells to produce, again, the desired product. And in terms of a lot of metabolic modeling projects going on around the world, Biolog data tends to be used as the truth to compare the predicted models against reality. And so this is from a paper by Bernard Paulson's group back in 2007, where they show how they cyclically go around this loop and keep improving their models, improve the predictability, challenge their predictability against Biolog data, Phenotype microarray data, and then again another round of improvement of their model. For the next section, I'd like to talk about applications having to do with gene function and genomics. The questions that we'll address include how you can determine the function of a gene, how you can improve genome annotation, and how you can even determine the function of an organelle. So in these kinds of experiments, we're typically comparing two different cells to see how they're similar, how they're different. So comparing a cell A versus a cell B, then run in the OmniLog collects the data, gives these kinetic graphs. We use two different colors. We use the red color for cell A, that being the wild type cell typically. Cell B would be the cell you want to compare it to, or typically a mutant with a gene knockout, for example. And then to do the comparison of cell A and cell B, the software will do an overlay of these two kinetic graphs. And wherever the red and green overlaps, it changes the color to yellow. So wherever you see yellow, this is where the cells are phenotypically behaving the same. And you see red if the red cell is giving a stronger response, and green if the green cell is giving a stronger response. That was just showing just one plate, but if you run the whole set of 20 plates, then you get a much more comprehensive picture, and you end up with a result as shown here on the right, where we compared two closely related bacteria. We see mostly yellow because in most of the phenotypes the cells were unchanged, but you see little black boxes that highlight wells with where there does appear to be an interesting relevant phenotypic difference. So start off here with a real example, a simple example with an E. coli bacterium, where we made a knockout of a gene called MALF, and this gene happens to be a transporter for the sugar maltose. So we expected when we knocked out this gene that we would see loss of metabolism of maltose. And in fact, that is what we saw. But in addition, we also saw loss of metabolism of maltotriose and dextrin, which happen to be the same sugar as maltose, but just a longer chain length. So this is telling us that the maltose transporter will transport all of these. When you knock it out, it loses the ability to metabolize these sugars. It recognizes the chemical structure of a maltose-like sugar, but it doesn't care about the length of the sugar. It's like sucking in a piece of spaghetti. It doesn't care how long that spaghetti is. It just sucks it into the cell. So you knock out MALF, you lose metabolism of these three sugars. Now in this case, we knocked out the gene by inserting a tetracycline cassette to disrupt the gene. And so in addition to a knockout, we have a knock-in of the tetracycline cassette, and we see the phenotypic changes that result from that because the cells become resistant to about 7 or 8 different tetracycline antibiotics that we have in our panel. So red indicates the phenotype that's lost. It's present in the wild type, missing in the mutant. Green is a phenotype that's gained, not present in the wild type, but present in the mutant. So maltose is lost and tetracycline resistance is gained. Now, there's a large number of genes whose function maybe we have a little information about or whose function is totally unknown. And this technology provides a direct phenotypic assay for gene function because you can just compare a knockout to a wild type and see which phenotypes change. So this is from a paper in 2006 from Sidney Kustu's lab at Berkeley, and this is a gene chip experiment with E. coli bacteria where they found an operon of completely unknown function, and what they knew was that these genes were turned on under nitrogen starvation conditions. So the strong implication was that it was a nitrogen-regulated pathway, but other than that, no information. So they simply made a knockout and ran a comparison of wild type, shown here, to mutant, shown below, with different nitrogen compounds, and they found that when they knocked out the pathway, the cell lost the ability to metabolize pyrimidines, cytosine, uracil, and uridine as nitrogen sources. And so pyrimidine catabolic pathways had been found in other bacteria and published previously. There were 2 other known pathways, but E. coli uses a different pathway, which they then discovered by using this Phenotype microarray approach. Another beautiful example of using this technology is this paper from 2010 from Joe Storino and Tom Wood's groups at Texas A&M at that time. And they were interested in asking a more subtle question, which is what is the role of phage genes, viral genes that are left over in the E. coli genome after the virus that brought them in is no longer active. And so in the E. coli K12 genome, there are 9 identified areas where a prophage had integrated into the E. coli genome. And then at some point in evolution, these could no longer form active viral particles, but there were still vestigial genes left behind from the virus. And the question is, why doesn't E. coli get rid of these genes? Could these genes be doing something beneficial for E. coli? And so just like doing a gene knockout, these researchers made precise deletions of each of these viral genes one at a time, compared them to the wild type, and they found, sure enough, that these genes provided a useful function to E. coli bacteria in terms of stress resistance. Some of these made the cells more resistant or tolerant of nalidixic acid and azlocillin, some antibiotics. They also found in one osmolarity tolerance. They found a tolerance of peroxide, acid, heat, and so on. So E. coli has decided to keep some of these genes because they provide some kinds of stress response relief. Another beautiful example from Cheryl Kerfeld's lab at Berkeley, and this is more of a genomic story. By doing genomic analysis and looking at different genome sequences, they found that there was evidence of a conserved gene cluster throughout the family of Planktomycetes that appeared to be some sort of a bacterial microcompartment, or what we might call an organelle. And so since they had identified these genes by doing a genomic analysis, they could also put in a mutation to knock out this gene cluster. And when they analyzed the knockout compared to the wild type, using phenotype microarrays, they were able to get a clue as to what the compartment was doing. And what they found in the knockout cells, the cells lost the ability to catabolize fucose and rhamnose. And what these carbon sources have in common is that they result in production of lactic aldehyde. Lactaldehyde is a very reactive compound. And so the implication was that they had this compartment to compartmentalize the aldehyde so that it wouldn't react and damage other proteins in the cell. And they found additional evidence to corroborate that proposal. In the next section now, some examples on environmental effects. Some typical questions: how does the nutrition of a cell alter its phenotypes and its biology? More specifically, what are effects of gases, carbon sources on the cell? Also, how do physical parameters alter cell phenotypes? What are the effects of specifically temperature, pH, salinity, and so on? So in terms of the effect of the gas environment of a cell, here's a simple experiment where we compared the carbon metabolism of E. coli either in the presence of oxygen aerobically, which is on the right, or anaerobically on the left. And I also like to show this as an example that we can run anaerobic bacteria and bacteria that require special gas atmospheres. Perhaps you can see in this slide that the plate on the left is actually inside of a gas-proof plastic bag with an oxygen-absorbing sachet. But you don't have to use sachets. You can also set these up and seal the bags inside of a gas chamber with any gas atmosphere you desire. But getting back to the point of this experiment is that the carbon metabolism of E. coli changes when it has oxygen available aerobically versus when it doesn't have oxygen available anaerobically. And specifically, what you see is that anaerobically, E. coli loses metabolism of carboxylic and amino acids. It still can metabolize sugars, but no longer can metabolize acids. Now, not only does the gas atmosphere regulate carbon metabolism in microorganisms, but carbon metabolism can regulate the nitrogen metabolism of the organism. Here I'm showing a side-by-side comparison of nitrogen metabolism in E. coli compared to a gram-positive bacteria, Staph aureus. Now, in the case of E. coli, it will use a large number of nitrogen compounds if you give it a poor carbon source like succinate. So, given succinate as a carbon source, it will use ammonia, it will use amino acids, amino sugars, purines, peptides, and so on. If we give it a carbon source that it likes better, which is glucose, it actually shuts off a lot of the pathways, still metabolizing peptides, but turning down the rate of metabolism and shutting off other nitrogen pathways in the cell. This is called the glucose effect, for example. And if you use something in between succinate and glucose, like glycerol or pyruvate, you'll get a pattern that's in between the succinate and the glucose pattern. So E. coli regulates nitrogen sort of like a rheostat, kind of up and down. But when we look at the gram-positive bacterium Staph aureus, we see a very different picture. It won't use succinate, so in this case we gave it pyruvate as the carboxylic acid carbon source, and we see that the cells will use some amino acids and peptides primarily as nitrogen sources. But if we give it a preferred carbon source of glucose, it's now primarily using ammonia, urea, and D-serine. This is not a subset of the pyruvate set, it's actually a completely different set. So Staph aureus, unlike E. coli, undergoes a much more drastic rearrangement of its nitrogen metabolism in response to the carbon source that it's using. Another environmental parameter that can have a profound effect on the biology of cells is temperature. And here we compared a temperature-sensitive bacterium, Yersinia pseudotuberculosis, which also is a pathogen. And we're looking at 3 different strains from our freezer collection. And now we're using the red and green colors to indicate temperature rather than different strains. So the red color indicates the metabolism at the warmer temperature of 33 degrees. The green color indicates the metabolism at the cooler temperature of 26 degrees. So we have a 7-degree difference here. And what we see primarily are yellow curves with a red shoulder, and that's indicating that the metabolism is occurring at both temperatures but faster at the warm temperature. And that's normally what you expect to see because thermodynamically the enzymes work faster and metabolism usually speeds up at warmer temperatures. However, we're seeing some very clear and drastic examples of pathways that are actually turned on at the lower temperatures. And each strain here behaved a little bit differently. The one on the right here was using, at the low temperature, glucose 6-phosphate and fructose 6-phosphate. The one in the middle is using glucose 1-phosphate and fructose 6-phosphate. And the one on the left is using also fructose 6-phosphate, but now some TCA cycle substrates at the low temperature. So we're seeing interesting strain-to-strain differences, and we're seeing consistently that metabolism of fructose 6-phosphate is turned on, and we think that this has importance in pathogenicity, which I don't have time to discuss. But I do have a section here now talking about applications in pathogenesis and epidemiology. And again, this is answering questions like, what are the characteristics of a pathogenic bacterium? How are pathogenic and non-pathogenic strains different? How do outbreak strains differ? And how is toxin synthesis regulated? So first, an example of a very important pathogenic bacterium, Mycobacterium tuberculosis, which was analyzed by Paul Wheeler's group in England, using phenotype microarray analysis. And first of all, you can see what the preferred carbon substrates of Mycobacterium tuberculosis are, and they tend to be fatty acids such as acetoines, acetate, acetoacetate. We can also differentiate and fingerprint different strains of Mycobacterium tuberculosis based on their carbon metabolism profile. So you can subtype the Mycobacterium using just the phenotype microarray plates. Second example here is a comparison of two outbreak strains of Vibrio cholera. The red color is the classical O1 strain, which caused most cholera epidemics up until the 1960s. And then the green color indicates the metabolism of the L-Tor strain, which arose in Bangladesh in the early 60s. And in less than a decade, it pretty much displaced the classical strain in terms of causing outbreaks. We see a lot of differences in their metabolic properties, but one that was particularly salient here was that we noticed that the classical strain did not grow very well unless it was supplemented with tweens in plate number 5 here. And what this is telling us is that the L-Tor strain does not require tweens for growth, but the classical strain does. And so we confirm that by just growing them on an agar medium, minimal medium. The classical strain without tweens grows very, very poorly. It grows much more strongly when you add tween, whereas the L-Tor strain is indifferent to the presence of tweens. So the classical strain will have a major growth disadvantage in an environment that does not have fatty acids available compared to the L-Tor strain, which could be a major factor or the major factor in L-Tor being able to outcompete. Here we have a comparison of two E. coli bacteria, the laboratory MG1655 E. coli K12 strain shown in the red color compared to E. coli 0157, a very pathogenic E. coli, in the green color. We can see the well-known phenotypes of 0157, that it's negative for sorbitol and that it's resistant to tellurite. And then we see consistently in a large collection that 0157 is consistently also negative for D-serine metabolism and positive for sucrose and N-acetylgalactosamine metabolism. Now, when the FDA group headed by Tom Sabula did a analysis of a large number of strains and then started looking at the genome composition, or the genomics of these bacteria, they found that the sucrose positivity and the N-acetylgalactosamine positivity correlated with the loss of D-serine metabolism, and they found out that this was because the sucrose and N-acetylgalactosamine metabolic genes inserted into the D-serine genes, knocking them out. However, as they look at outbreaks, they occasionally do find a variance. So this is from a spinach outbreak from this, again, Amit Mukherjee, Tom Sabula at the US FDA. And they were finding unusual mutants or isolates that had lost metabolism of N-acetylgalactosamine, both for carbon and nitrogen metabolism. And in New Zealand, they found sucrose negative isolates, particularly in the South Island of New Zealand. This is the predominant 0157 that they get. They first saw it in the Biolog plates, and then they could replicate that with fermentation assays and throughout a large culture collection. This is a paper recently published by Roberto Laragione and collaborators in England. Campylobacter is another important pathogen. Coli and jejuni are two different species that are difficult to differentiate because they're quite similar. But they found a nice correlation here where they could differentiate them in the Biolog phenotype microarrays based on metabolism of propionate. The coli strains are strong metabolizers of propionate, whereas the jejuni strains are negative or weak for propionate metabolism. And here another nice example from Carol Maddox's group at University of Illinois. This is a veterinary example where they were able to distinguish a vaccine strain from pathogenic wild strains of Strep equi based on their metabolism of two carbon sources, psychose and methylpyruvate. And also quite exciting is the use of the phenotyping technology to determine conditions that turn on toxin production by pathogenic microorganisms. The first breakthrough here was made by Gardner and colleagues in Australia. They were working on a fungal pathogen called Fusarium graminearum, which produces a mycotoxin that kills wheat. So agriculturally, this is a very important fungus and very bad for agriculture. However, even though this Fusarium is very good at producing toxin in the field and killing wheat, when you bring the fungus into the lab and culture it under laboratory conditions, the toxin genes are tightly shut off. And over the many decades of research, nobody could ever figure out what are the magical conditions that turn on toxin production. And so they approached this problem by making a green fluorescent protein fusion to the promoter of the toxin genes and using the phenotype microarray plates to test hundreds of different culture conditions to see if they could detect turn on of toxin production. So they set up an experiment and followed for 7 days and looking for green fluorescence in the wells of the phenotype microarray. And nothing happened up until the third or fourth day. And finally, on day 3 or day 4, they started to see green fluorescence in 4 wells, here, here, and here. And it turned out that it was the use of polyamines such as arginine, putrescine, agmatine, and guanine as nitrogen sources, which are the strongest factors that turn on toxin production in Fusarium. We were inspired by this work and wanted to see whether we could generalize this and make this work in a bacterial example. We picked a very important but difficult one, Clostridium difficile. We cultured the Clostridium difficile again for 3 days in the wells of phenotype microarray plate. We transfer it through a filter step so that we can separate the supernatant, and then we transfer 5 microliters of that supernatant onto a third microtiter plate where we have a sensitive mammalian cell line. And if there's toxin present, it kills the mammalian cells. They produce less energy, and you see a light purple color in the well. So here's a close-up of that particular plate, and we detected toxin production. In this particular plate happened to have a lot of arginine peptides. So just as arginine was an inducer of toxin production in the Fusarium, and also we found it to be a toxin inducer in Clostridium difficile, but we found things that actually gave even higher induction than arginine. And finally, I just want to talk about the applications of using 2,000 culture conditions and looking at microorganisms, allowing you to address questions like how do cells change under different culture conditions, how do such changes impact their biology, and how does this analysis provide a more complete picture of the true complexity of a microbial cell. And so this is a concept slide, but very important, and I think people frequently fail to think about this. But microorganisms are always sensing their environment, and when they sense a change in their environment, they remodel themselves to try to live more appropriately or survive more appropriately in that particular changed environment. Every time even you make the slightest change in the environment of a cell, whether it's nutritional or physical change or some other chemical change, the cell will adapt. It'll turn on genes, turn off genes, make proteins, change its membrane, and so on. And so every culture condition of the cell, the cell remodels itself and literally becomes a different cell in every environment. Literally and physically becomes a different cell. You might think that the change might be fairly small and trivial, but here's a beautiful example to disabuse you of that opinion. Here we have Saccharomyces on the left, an electron micrograph of what it looks like when it's cultured on glucose as a carbon source, and on the right, what the same cell looks like when it's just cultured on a fatty acid as a carbon source. The organellar composition is drastically altered. On glucose, the cells are fermenting. They don't need mitochondria very much, if at all. When they are growing on oleic acid, they have a lot more mitochondria. They're making peroxisomes because there's peroxide formed from oxidation of fatty acids. These are probably little globules of uptake of fatty acids. Their membrane and the cell shape is altered as well. Another fungal example, Candida albicans, which can grow either in small single cell stage, or they can shift to growing into a filamentous growth stage. And this is actually very important for human health because the single cell form is in the non-pathogenic form, whereas the filamentous form is the pathogenic form. And so understanding what triggers this morphological conversion will also give us insight into how to prevent the pathogenicity of Candida albicans. So the phenotype microarray then is, you can think of it as a way to take a cell that you've grown in one environment, and by putting it into the phenotype microarray, you're now exposing that cell to 2,000 different environments, and in each environment the cell changes, and so the phenotype microarray is allowing you to see 2,000 faces of that cell. And you can address interesting aspects of biology of microorganisms, for example, growth rate and survival, sporulation and germination, biofilm formation, expression of surface antigens, ability to host viral growth, secondary metabolite production, and toxin production, as we've just illustrated. So this phenotyping technology has been available for over a decade now. Over that decade, we've evolved protocols so that this technology can be applied to an extremely wide range of microorganisms. The typical current uses are for analyzing genes, analyzing and comparing cell lines, determining the biological properties in detail of a particular cell, analyzing effects of chemicals on cells, which I didn't get much of an opportunity to talk about, and improving bioprocesses. Some of the newer uses of the technology are in microbiome studies based on phenomics rather than metagenomic sequencing, or in addition to metagenomic sequencing, and analyzing human cells and interactions of human cells with microbes. So in my last slide, I'm just showing in the plate on the right that this technology now can work. We have a way to get this technology to work also with human cells, so we can do the same kind of phenomic analysis of human cells, and you can compare uninfected versus infected human cells. And I'm just showing you here a human colon cell run side by side with an E. coli bacteria. This is in the phenotype microarray plate that's designed for the human cells, but the bacterial cells can also be run in this plate. And these two bacteria live in the same environment, which is the human colon, and yet they have obviously different metabolic properties and different phenotypic properties. And so we see a very interesting future expansion of the phenomic technology with not only microbial cells, but now with human and animal cells as well. So that's the end of my presentation. Thank you very much for listening.